past Tuesday, organic students entered a lab and received an unknown by Dr. Mullins. Our goal for the lab period was to determine the unknown. Students were also given a list of knowns that our unknown could possibly be. Our goal was to find the structures and the melting points of what the knowns could be, and then run a series of tests on our unknowns to determine what our unknown might possibly be. The first test that I ran on my unknown, which was labeled AL, was a melting point range. I first received a small portion of my unknown into a dish and pushed it into a capillary tube. After balancing the capillary tube down a glass rod to make sure the contents were at the bottom of the capillary tube, I put the capillary tube into the mel temp instrument. At the end of the mel temp test, I found out that my melting point range for my unknown was 169 degrees Celsius to 171 degrees Celsius. My unknown also looked like a fine white powder to the unaided eye. Another test that I ran on my unknown was an IR spectroscopy. I did this to see, I ran my unknown through the IR to see what functional groups could possibly be in my unknown. Again, putting a small sample of my unknown onto a dish, I also added a small portion of KBR and mixed it with my unknown. After I did that, I put it into a mortar and pestle and smashed it into a fine white powder again and again so that I was able to put it into a small bolt. I squeezed it until it was a small pellet and then put that on a IR and ran it through the IR spectroscopy. My results from the IR were a broad spectrum of OH alcohol groups starting at 370 and ended at 2450. Based on these two results, I was able to eliminate many of the knowns that the unknown could possibly be. The test that I ran eliminated the knowns based on their melting point ranges, temperatures, and the functional groups that it could be. If it did not have any alcohol functional groups, or if it was not in the appropriate range for the melting temp, then the knowns were eliminated. This also eliminated any knowns that had a close melting range to my unknown that did not have any alcohols as their functional groups. After eliminating all the knowns that my unknown could not be, I found out that my known was mannitol. The mannitol has a melting point range of 165 degrees Celsius to 169 degrees Celsius and has 7 functional groups as alcohols. It is the only functional group present in mannitol. And that is how I came to my conclusion of mannitol. If I was able to redo this experiment, then I would make sure that maybe, since my melting point range was just a few degrees higher than what the mannitol's melting point ranges were, the one conclusion that I could come up as to why this happened was maybe that I had too much of my unknown in the capillary tube. This would have caused the melting point to range since there was so much in the capillary tube to melt. Another thing that I would change is I would run more than just one IR spectroscopy for my unknown, just to have more IR graphs, just to make sure that the first one that I ran was correct and that there was no impurities in my IR group. So when given an unknown in the lab, the series of tests that we ran on it were a melting point range and a IR spectroscopy on the unknown. Another test that I could have done was to see if it was soluble or insoluble in water, but that test was not run on my unknown. And those were the series of tests that led me to my conclusion that my unknown, which was labeled AL, was actually found to be mannitol. Thank you for watching.